Hi, I am Blanca Limones from IMDEA Materials Institute, and this is BIOMET4D Science Explained. In this video, we are going to talk about a specific process called Inductively coupled plasma optical emission spectroscopy, or in this video, ICP for short. This is a powerful analytical technique which tells us exactly which elements are present in our solution and how much of each. As part of the Horizon Europe BIOMET4D project, we are analyzing elements like zinc, magnesium, nickel, titanium, and yttrium in the development of materials for biodegradable metallic shape-morphing implants. In cell culture research, you study how materials interact with biological environments, how cells respond, how materials degrade, and how they might perform inside the body. We currently apply this technique for different purposes: to determine baseline atomic concentration in organs before material implantation, to analyze simulated body fluid solutions following material degradation, and, as we will discuss today, to monitor elemental concentrations in cell culture media during the degradation process. Each day, we collect the samples to analyze how the concentration of elements has changed as the material degrades. This is usually done with acid digestion, and we normally use nitric acid solutions. All our samples are then diluted in order to obtain a clear, homogeneous solution suitable for introduction into the instrument. Before continuing with the process, a series of standard solutions with known elements concentrations are made. These are used to create a calibration curve to relate emission intensity with concentration. Once we have all the samples that we are going to test, we need to create what is known as a lab book to inform the ICP about the location of each sample and their respective dilutions. This outlines all of the element specification to be analyzed. After that, the rack is simply placed in the sample reading position to start the measurements. It's at this point that the injector takes the sample, which is drawn into a nebulizer, which converts it into a fine aerosol mist. Now, here comes the exciting part. The mist enters a small glass torch where a powerful plasma, a super hot, glowing gas, is waiting. This plasma, made from argon, burns as hot as 10,000 degrees Celsius. Inside it, every droplet of our sample is vaporized, atomized, and energized, turned into individual atoms that start to glow with energy, released as light at wavelengths characteristic of each element. The light from the plasma then travels into a spectrometer. It splits the light into its different colors and measures how bright each one is. The brightness tells us how much of each element is in the sample. Pretty cool, right? Finally, the computer compares those colorful signals with the known standards and produces a report. At this point, we need to do some corrections to the signals before analyzing the data, but that's a topic for another video. I am Blanca Limones from IMDEA Materials Institute, and this has been BIOMET4D Science Explained. If you enjoyed the video, do not forget to like and subscribe.